Hello and welcome to the last part of genetics that we are going to discuss today over here. In the last few classes, we have discussed the entirety, almost the entirety of the genetics part that we were supposed to do. But now we are left with a very few things that are left to be discussed. We will be discussing those things today, and with that, we will be finishing off the chapter of genetics. Right? So, am I audible? Am I audible? Just let me know if I'm audible or not, or everything is working fine or not, and then I will start with the remaining parts of the genetics chapter. And with that, we will be finishing off this chapter. Very few things are left. We will be doing that, and this is the last installment of the chapter of genetics, right? Hello, am I audible? Okay, right. So let's deal with the idea of incomplete, not inheritance. It has to be incomplete dominance, right? Let's come to that. It is we are going to talk about is incomplete dominance. Incomplete dominance, right? Now, to understand the idea of incomplete dominance, we have to go back to our primary understanding of dominant and recessive. So when we were dealing with the idea of dominant and recessive, we took one very handy example of the paint buckets, right? So when we took the example of paint buckets, we took one example, that is, suppose I have one paint bucket over here. Suppose this is one paint bucket that I have, and this is another paint bucket I have. Now this paint bucket over here contains black color, and this paint bucket contains red color. Same quantity, identical containers, or identical buckets of paint. Now, what happens when I mix these two? Now, when I mix these two, the resulting paint that results over here ultimately looks something blackish, right? Whenever you add them, it looks something blackish red, or it's very dark or darkish red. So ultimately, the blackness prevails, right? So from that point of view, we were able to say that the black paint over here was dominant, because after mixture also, the characteristics of the black paint, that is being dark and being very shadowy shade, was the dominant feature or the prevalent feature. But the prevalence of redness in the resultant paint was absent, right? So over here we were able to say that the red paint was somehow recessive. Can you recall this bucket theory that we tried to that I tried to introduce as an analogy of dominant and recessive, right? Okay, is it understandable? Right. So now basically this analogy, if you are able to understand this analogy, then we will bring back another concept to it. Bring things, we will add up things over here. Now this is one kind of idea. Over here we had the idea of dominant and recessive. But all it will not always be the case if we don't mix colors like black and red. Now what we can do in the later part of the discussion, we can add two different colors, like Suppose I have another bucket and two more buckets. Now in one bucket I have the red paint, and in another bucket I have the white paint. Now to represent white, I will be using over here the color yellow, because that, or gray basically, because that's the closest color I have resembling white over here, since I cannot make white on a whiteboard. So let's take a red and white colors, and now if I mix these red and white colors into a large container over here, now in that large container what we are supposed to get, we are supposed to get a color which is pink. We will be getting a color which is pink, right? We will get something known as pink, right? Now in the pink color, both the characteristics or the features of both red and white colors are present, right? Over here both redness and whiteness are prevalent. Actually it's not that both redness and whiteness is prevalent. Over here we can say it like this: we have redness present but not red enough to match the red paint, but not red enough. But not red enough. So pink color is a variety. Yeah, both have distinctive impact over here, and due to that impact what we have a color which has a redness in it, but it's not red enough to match with the original red pen. It's not matching, right? So what we can say over here, suppose if we draw a line, like in mathematics we have learned about a number line, right? So similarly if we draw a line over here, suppose this is a line over here, in that line we mark this end is our absolute red, and this very end over here represents the end where we have the white color, right? Now after crossing the two conditions, after mixing, the resultant color or the final color that we are getting, that's neither white nor red, but it's something in between red and white, isn't it? So it's something in between red and white. It's something in between red and white, which is pink. So what happened in the resultant mixture? What happened? The color tried to move towards the dominant one, right? The color tried to shift towards the dominant one. As in the previous case, what happened? The entirety of the color in the resultant solution represent or tended to be similar to black. So it tried to stay on the side of the black. Now over here, the resultant color also tried to be on the side of the dominant one, that is the red one. But in its journey, it got stuck in between. So in its journey of becoming dominant is incomplete. Can you see that? Can you appreciate the fact? So journey of becoming dominant is incomplete. Is this understandable? Are you on the same page? Over here also, as a result of mixture, the color tried to shift towards the dominant side of the spectrum. But in that effort, it got stuck in between the red and white. That means it was trying to move towards the red, but it was not able to move towards the red entirely. It got stuck in between in a color like pink, right? But in the previous situation, what happened? In the previous situation, the black was the dominant one and the red was the recessive one. So the color moved from white towards black, but it ended up being near about, in a place near about black, right? So it was almost completely dominant. The final one showed dominant traits. But over here, the final resultant mixture is not able to show dominant traits because its journey of becoming dominant got stuck in between in a color which is more or less intermediate, right? Which is pink. Now, are we clear till this? Are we clear with this idea of bucket hypothesis or bucket theory or bucket analogy? Right. Now, over here, try to appreciate another thing. Right. So what happens over here? Another thing that if I try to make, if I try to say, that will be, see over here, the color is intermediate between red and white. So this kind of inheritance pattern is also known as intermediate expression of gene. Intermediate dominance is also a term that is used. Intermediate dominance. Right. Intermediate dominance. Okay, because the resultant one is becoming intermediate. Now, remember this analogy of red, white, and pink. Why I'm telling you to remember this? Because there's a plant known as Mirabilis jalepa. In that plant, what happens? The dominant flower color is red. I mean, dominant petal color of the flowers is red. The recessive color of the petals is white. Now, if we cross them in the F1 generation, we get pink petaled flowers. Some of them are pink petaled. Right. Now, how can we represent this in a cross? Right. Let's understand that cross of incomplete dominance or intermediate dominance. Now, before going to that, I would like to show another thing over here to you. That is, suppose we are mixing. Suppose I have a can full of, I have two cans of paint. Now in one can I have can full of red paint and another can I have can full of white paint. Right. Now if these two cans, when I mix these two cans, if these cans give me a paint where we have red and white alternatively, like a 2-in-1 ice cream that we get in the ice cream shops, or a polka dot kind of pattern, dots of red in white or dots of white in red, red plus white situation, red plus white situation, that situation can be one case. Another case, it can be a full can full of pink color, right? It will be pink. Now if this is becoming pink, then that situation is called incomplete dominance or intermediate dominance. And if both red and white are present together in the resultant mixture, that will be called as your codominance. We learned about codominance in the last part, right? The immediate last, isn't it? Are we clear till this? Right. Now we will go and check the monohybrid cross for a incomplete dominance. Is it okay with you? Is it okay? So let's see monohybrid cross for a incomplete dominance, right? Over here, suppose over here what we have a plant known as prime rose or 4 o'clock plant. Right. In that plant, what I discussed, the flower color, this flower color, the petals of the flower, if they are red, red petals are dominant and white petals are recessive. White petals are recessive. Recessive is white petals. Right. Now let's mark the gene, the dominant allele, to be capital R for red and small r for the recessive one. Right. So suppose let's take the male. Let's say the male is a white flower. So if it has a white flower, so it will be small r, And if the female is having a red flower, suppose the female is having red flower, so the genotype will be capital R, capital R. So this is your parental generation. Now I'm not drawing the gametes over here. Okay, I'm waiting for you. I'm going a bit slow. I was picking up pace. Just let me know when you're done taking notes and assimilating the concepts that I discussed till now, right? Then I will move forward with the concepts. Right. So let's proceed further. So we have a plant known as a prime rose plant or 4 o'clock plant. Now, what happens with that 4 o'clock plant? This plant has red petal flowers as well as white petal flowers. Now, the red petals of the plant represent dominant allele of a gene which is responsible for the petal color of the flowers, and the white petals represent the recessive allele of the gene which is responsible for the flower color in this 4 o'clock plant or primrose plant. Right. Now we want to do a monohybrid cross with this gene for petal color. Now we are taking a male which has white flowers. If the male has white flowers, that means the genotype of the male will be smaller, that means recessive. And we take a female which has red flowers. So as the female has red flowers, so it will be dominant, that means capital R, capital R, right? Now just tell me what will be the F1 generation. I'm not going into the gamete writing and all that, because we know, I know that you already have learned enough of monohybrid cross, right? So what will be the genotype of the F1 generation? Just think about it and tell me what will be the genotype or genetic configuration in F1 generation. So the genotype of the F1 generation is supposed to be capital R, small r, isn't it? From over here we will have one of the R's coming in from over here and one of the R's coming in from here, right? So all the 4 individuals will be capital R, small r, right? Now for the F2 generation we will be doing what? We will be doing a selfing, and with that selfing what will we do? We will go to a Punnett square. So if we go to a okay, now what will be the phenotype of this capital R, small r? Capital R, small r will be, what will be the phenotype? The phenotype will be pink, because see, one allele for white, one allele for red, together they will make pink, because this plant shows incomplete dominance. Right? Now if we do a Punnett square, what it will show? Over here, suppose this is male, this is female, capital R, small r, capital R, small r. It will be capital R, capital R, capital R, small r, capital R, small r, small r, small r. Now out of them, this one will be red, this one will be white, the rest two will be pink. So over here, both phenotypic ratio as well as genotypic ratio will be 1:2:1. So what is the conclusion? Phenotypic ratio, which is equal to genotypic ratio, which is equal to 1:2:1. Exactly, right. So are we clear till now? So what you have to remember, what is incomplete dominance? Incomplete dominance, the color or the character will be intermediate between the dominant and the recessive in the F1 generation. Now when you do cross the F1 generation, selfing of the F1 generation, you will get F2 generation. Now in F2 generation, the phenotypic ratio and the genotypic ratio will be 1:2:1. Both the phenotypic ratio and genotypic ratio will be same. So what kind of questions can be asked in the exam? Now in dash plant, phenotypic ratio is equal to genotypic ratio. Now there will be options like 4 o'clock plant, 5 o'clock plant, pea plant, this rose plant, etc. So you have to remember this name of this plant because this name is particularly mentioned in your syllabus, that is 4 o'clock plant or prime rose, right? Are we clear? Now this is the point to be, whatever I explained to you about incomplete dominance, that you have to keep in mind the concept of incomplete dominance. You have to remember where it is seen, that is 4 o'clock plant, and you have to remember what is the conclusion of the monohybrid cross in our incomplete dominance, that is phenotypic ratio is equal to genotypic ratio is equal to 1:2:1. And what is that 1:2:1? 1 represents red, 2 represents pink, and 1 represents white, right? Understood? With this much of understanding, let's proceed on into the inheritances, right? Different form of inheritances. So what we are going to learn right now, right, over here, let's start with, we have many chromosomes. You already know the concept of chromosomes, right? Now these chromosomes are of two types. Chromosomes can be autosomes or they can be allosomes. Allosomes are chromosomes which govern sex or sex governing chromosomes. Right. And autosomes are those chromosomes which do not govern sex, which are common in both male and female, right? Now what are these chromosomes? So now if we take the example of humans have 44 plus XY or 44 plus XX, this kind of arrangement. They have 46 chromosomes, right? So now if this is the situation, then autosomes will be the 44 chromosomes which are exactly arranged in 22 pairs. And what are the allosomes? Allosomes will be X chromosome and the Y chromosome. Now are we clear till now? Okay, are we clear till this? Right? Try to assimilate this much. Is it alright? Have you understood this much? Now, now let's come to the idea. Suppose over here, suppose we have a gene A. For example, take one gene, we have A gene. Now in A gene, we will have two alleles. Okay, two alleles. Now, out of those two alleles, what will happen? There will be one capital A, one other one will be small a. Now over here, if we have the arrangement like capital A, that is known as homozygous, or even small a. Or if we have capital A small a, that is known as heterozygous, isn't it? We know all this, right? Right. Now, but suppose this kind of situation can also be said when we have the XX arrangement. But when we have XY arrangement, we have two different chromosomes. Two different chromosomes. And each gene has only one allele. Each gene on either X or Y has one allele, which is controversing the popular belief that we had till now. Right, isn't it? All right. Are we clear till this? take some time and try to assimilate the concept over here. Over here, try to understand that whenever we used to have homologous chromosomes or homologous pairing. Over here, these 22 pairs represent the homologous pairing. But over here, whenever you're the XY situation, you cannot have a homologous pairing because you have two entirely different chromosomes, right? One is the X chromosome, another one is Y chromosome. Right, isn't it? Just take some time and try to assimilate this. This XX arrangement is present in females and XY arrangement is present in males. XY in males, right? This is the situation in case of mammals. Now, if we go down into grasshoppers or uh, chickens, then you will see the things are different. The chromosomes, allosomes are different actually. Now, autosomes are more or less same in all the animals, but the allosomes differ, right? Okay. So now, this condition of having only one allele, one of the homologous pair chromosomes and only one allele, this situation is known as hemizygous condition. Hemizygous condition. So hemizygous condition is only seen in case of human males who have XY situation, right? All right. So if we are clear with this much of knowledge, or if we are done assimilating this much of knowledge, let's go further. Which part exactly you want me to repeat? Okay, so basically we discussed nothing but we have chromosomes. We all learned about chromosomes. Okay, so the chromosomes can be of two types. They can be either autosomes or allosomes. Now, allosomes are those chromosomes which govern the sexual characteristics, right? Sex governing chromosomes. And those chromosomes which do not govern the sex, they are known as autosomes. Now, in human, if we see the chromosomal arrangement, we can see that it is either 44 plus XX or it is 44 plus XY, right? So, now out of those, the 44 that we see is the number of autosomes, right? And those autosomes are all have homologous pairing. That means those are nothing but 22 homologous pairs, right? That means if they have homologous pairs, that means each gene on them will have two alternate alleles, right? That means if I have one gene A, then that gene will have two alleles, that is capital A and small a. Is it clear? Now, if the gene has two different alleles, capital A and small a, then the small a and capital A arrangements will be known as homozygous, whereas the capital A small a arrangement will be known as the heterozygous. This much we have already learned about autosomes, right? Now we come to the concept of allosomes. Allosomes are what? Allosomes are sex governing chromosomes. That means the chromosomes which govern the sexual characteristics. Now, in humans, we have two such chromosomes. One is the X chromosome, another one is the Y chromosome. Now, you have two possible combinations of these. You can have either XX possible or you can have XY possible. There's no such combination known as YY, right? Now, the XX situation is present in the human females and the XY situation is present in the human males, alright? Now, over here, if you see that we talked about homozygous and heterozygous. These homozygous and heterozygous concepts are only applicable whenever you have the homologous pairing of chromosomes. That means similar kind of chromosomes, one coming from mother and one coming from father, pair up in your body, having same locus. In the locus they have same gene and all that stuff. That is possible for all the autosomes because autosomes are nothing but 22 homologous pairs, and that is possible for the females because the allosomes in the females are also XX. That means they have a homologous pairing in X chromosome as well. But this situation is not possible in case of human males. Why that is not possible in case of human males? Because in human males the allosomes have a XY arrangement, right? Now this XY arrangement, in that XY arrangement, even though we are writing them as a pair, they do not form a homologous pair. They are non-homologous pair, right? Because the chromosomes are entirely different. X is an entirely different chromosome, whereas Y is another entirely different chromosome, right? That means the genes that are present on the X chromosome and on the Y chromosome in a human male, all those genes have only one allele in that particular human, because X chromosome does not have its homologous pair present, and similarly Y chromosome does not have its homologous pair present. That means all the genes present on X chromosome as well as Y chromosome in a XY situation, that means in a human male, are having only one allele. And this situation of having only one allele to represent one gene is known as hemizygous condition. So this is what we discussed so far about chromosomes. So this is the idea of hemizygous condition. Now are we clear? Are we good to go? Right. Now since we have understood this idea of hemizygous chrome, now from here we can deduce few simple things, right? Now over here, if we see, you have to just remember what is hemizygous condition. So now after all this discussion, we have three conditions that we can see. We have one known as homozygous condition, another is heterozygous condition, and the third is hemizygous condition. So previously you only learned about homozygous and heterozygous. Now there was one small addition into that, that is hemizygous, right? Now let's deduce few things from this discussion. Now if we say that, or we have already learned that females have allosomes XX and males have allosomes XY. That means we can say that X chromosome is present in both male and female, both male and female, but Y chromosomes are only in male. Can we deduce this, or can we draw this conclusion from our understanding of allosomes? Can we draw this simple conclusion from our understanding of allosomes? That is, what is the conclusion? X chromosomes are present in both male as well as female, whereas Y chromosome is only present in male. Right. Now we have some characteristics which are known as holandric. What is that? There are certain genes on Y chromosome. Y chromosome has certain genes on it, right? And if Y chromosome is only present in male, that means this Y chromosome can be passed from father to son. Mothers cannot pass it to sons, and from son to grandson to great grandson. Isn't it? Right? Isn't it? So the Y chromosome can be only passed on by males in the family. That means the father can send it to son, son can send it to grandson, and grandson can give it to the great grandson. Alright, there's no role of females in the inheritance of Y chromosome. Females have nothing to do with the inheritance of Y chromosome, isn't it? Is this conclusion correct? Is it okay to draw this conclusion? Right. Now, the genes which are present on the Y chromosome, they are known as, that's why, holandric genes or holandric, and the traits expressed by those genes are known as holandric traits. Holandric traits. So what is the meaning of holandric? Andric means male, whole means completely. So these are completely male traits or completely male characteristics actually which have traits. Holandric character, we can say characters. The better term is to use characters. Holandric characters. Right. Holandric characters. Now this term holandric is very much important, that you have to remember. Holandric term is very important. Right. Now what are the holandric characters actually? Like you will see the pattern of hair distribution in the body. Now some males have inherently high amount of hair growth in their body, like very thick beard, mustache, chest hair, etc. Whereas certain males have very scarce body hair, very scarce beard or mustache. So this pattern of male hair growth, these are all known as male hairs because chest hairs or beards are not seen in females. Now these things are governed by some Hollandric genes. So these are conserved in a family. Like the pattern of hair growth on the body will be similar in a father and a son, and that will be same in a grandfather and a grandson, right? Another thing what happens, like there's hair growth on the pinna or the ear, external ear. Some males show hair growth on the external ear. That is also an Hollandric condition. The external appearance or the phenotypic appearance of the male genitalia, that is scrotum, penis, etc. These things also resemble or are Hollandric. That means the phenotypic appearance or appearance of the genitalia of the father will resemble the genitalia of the son, and that will resemble the genitalia of the grandson. Right. So these are Hollandric characters. All right. So this is, and the inheritance of Hollandric characters is known as Hollandric inheritance or Y-linked inheritance, because inheritance of Y genes. So this is known as Y-linked inheritance. Right? Is it okay? Right? Take some time and assimilate this much of knowledge. Now, if this much is clear, then we will move forward. Right? If this much is clear, then we will move forward. Right? So let's moving on. We have what kind of inheritance patterns we have. Now we come to the idea of inheritance patterns. Inheritance patterns. Now inheritance can be of two types: autosomal inheritance, that means inheritance of the genes present on the autosomes, and the inheritance can be allosomal inheritance, that means genes present on the allosomes, that is X and Y. Now allosomal inheritance again can be of two types: X-linked inheritance, that is the inheritance of X-linked genes, that means genes present on X, and the inheritance of Y-linked genes. Now the inheritance of Y-linked genes is also known as Hollandric, that we discussed just now. Hollandric. Right? Now over here, if there's a disease, right? Suppose there is a disease, and the disease lies in the autosomal dominant allele, so the inheritance pattern will be autosomal dominant, and the inheritance will be autosomal. If it is on the recessive gene, it will be autosomal recessive, right? Autosomal dominant and autosomal recessive. Similarly, if the disease is on the X chromosome, on some gene in the X chromosome, so it can be X-linked dominance, X-linked dominant, or it can be X-linked recessive, right? Are we clear with this? These are the inheritance patterns. Okay, take some time and assimilate this much of concepts. Are you done taking down notes? Okay, all right. Now, thank God we do not have much of Y-linked inheritance, autosomal dominant, or autosomal recessive in our syllabus. We only have two Mendelian disorders, X-linked recessive Mendelian disorders in our syllabus, right? We only have two X-linked Mendelian disorders in our syllabus, which are, we do not have to delve deep into the idea of autosomal dominant inheritance pattern, autosomal recessive inheritance pattern, or X-linked dominant inheritance pattern, or Hollandry inheritance pattern. We just need to know what is X-linked recessive inheritance pattern and two diseases linked to that. Now, what are those two diseases? Those are very commonly heard diseases. Those are hemophilia and red-green color blindness. Now this, sorry, it's blindness. Now this thing is commonly referred to as color blindness, but as a student of biology you should always know that there are different type of color blindnesses present. There are several color blindnesses which are present in the human population. Now which one is X-linked recessive is red green color blindness. The inheritance pattern of both the diseases are extremely similar. Both are X-linked recessive. So I will be showing only one inheritance pattern and I will be discussing one or two things about the diseases, right? Now starting with hemophilia, if I start with hemophilia, then hemophilia is what? Hemophilia is a clotting disorder, bleeding disorder actually. Now this occurs due to deficiency of certain clotting factors, clotting factor 8 and clotting factor 9. Right, so deficiency of clotting factor 8 and or 9 leads to hemophilia. Let's come to that. This thing you are supposed to remember, that is deficiency of clotting factor 8 and or 9. We do not have details of clotting factors in our syllabus. And just remember that clotting factors are always written in Roman numerals, right? We do not use English numerals of 8 and 9. Okay, so deficiency of clotting factor 8 or clotting factor 9 or both clotting factor 8 and 9, that leads to a bleeding disorder which is known as hemophilia. Now hemophilia is known as a royal disease or royal disease because it was a problem in the family of royal family of England, and boys in the royal family of England suffered hemophilia, and that's why the queens took the lead and queens started to rule the country. That's why we are so obsessed with, or the country is so obsessed with, the Queen of England, and there's no mention or there's no much talk about the King of England per se, right? Because the boys for a long period of time in the royal family of England suffered from this disease of hemophilia. Later on, with proper counseling and proper cure and proper genetic consultation and manipulation, the disease was removed or properly managed in the family, right? People were able to do away with the disease of hemophilia in the royal family, right? And this is the basic idea of hemophilia. And what is the idea of red green color blindness? Now there are certain males which are there, and they fail to distinguish the red and green color. The red and they have a colored vision. It's not like that. Many people think they have color blindness, that means they will have a black and white vision. It's not that they have a monochromic vision. They have a polychromic vision, that means colored vision. They just cannot distinguish between red and green color. Red and green color appear somewhat to be purplish to them, and they appear more or less same. So they cannot distinguish red and green color, right? Now there's an interesting trivia about red green color blindness. Now previously red green colorblind people were not allowed to drive. Driving was not allowed in red green colorblind people because they won't be able to distinguish the red signal with the green signal, so they will lead that would have led to accidents, right? That's why they were barred from driving, they were denied driving licenses. But later on there was a change in the traffic lighting system. What was that change? Previously, I hope you have noticed, and still now in many parts of India the traffic lights are somewhat like that: red, green and yellow. This kind of traffic lighting system, right? This was the previous pattern of color traffic lighting. But later on, to allow the colorblind people to drive, what was done? So this kind of situation was created. What was the situation? This was made for green, yellow was left untouched, this kind of pattern, and red was made like this. Red was made cross, right? So now this kind of pattern, like this kind of signal boxes are visible nowadays, right? This pattern is obsolete. Still now in different parts of India you can find this kind of patterns, but this is the standard pattern now. And this pattern allows the red green colorblind person to differentiate the red light with the green light. Now they have a clear distinction whether the signal showing a go arrow, that is arrow, or a stop cross, right? So this was the idea of red green color blindness, and why males are only affected, that we will be understanding while we will be seeing a cross. Okay, so let's see a monohybrid cross. Right, suppose over here it is X-linked recessive, right? So now let's take the healthy allele to be capital X and the diseased allele to be X star. We add a star at the head of the X. This is the standard notation in case of X-linked recessive diseases, right? Now what happens? We will have a father. Now suppose the father is colorblind. Suppose the father is normal. So father has a normal X gene, normal Y gene. So father is normal or not colorblind, color normal or non-hemophilic. Marries one mother who is a carrier for colorblindness, right? So over here let's go through these conditions. This is normal. Suppose one star you have, suppose you have a star on one X chromosome, that means you have one diseased X chromosome, one non-diseased X chromosome. This situation is known as what? Carrier of the disease. And if you have both the X chromosomes affected, then you have a diseased condition, diseased, because it is a X-linked recessive condition, right? You can have a recessive genes expressing when both the recessive alleles are present, right? Isn't it? Are we clear with this? And in case of X-linked inheritances, this situation is extremely rare. Diseased females are extremely rare, because to give birth to a diseased female you have to have a diseased father as well as at least a carrier mother, right? Now let's do the cross. If we do the cross over here, what kind of situations we are going to get? Suppose this is the parental generation. Now what will be the gametes? Gamete can be either X or Y in case of a male, or it will be a normal X gamete in case of the female, or a diseased X in case of the female. Now if these gametes meet each other, what kind of kids will be formed? Let's see. If these two meet each other, capital X, normal individual. Now if these two meet each other, then capital X, capital Y, normal individual. Now then if these two meet each other, capital X, normal individual. But when you're coming to this combination, that is capital X starred and Y, this combination is a diseased combination. Now see over here, only one recessive allele, right? You have only one X chromosome, and in that one X chromosome also you have the only gene that is responsible for color blindness or for causing red green colorful vision is diseased. So what it will lead to? It will lead to diseased condition. This is diseased, right? Now over here we do not do a F2 generation, we just stop till F1 generation, right? This is what people may ask you to do. That's it and nothing else. People will say a person has a normal father and a carrier mother, then what is the probability of a diseased child? Now what is the probability of giving birth to a diseased child? 25%, because out of 4 children, 1 child is diseased. Now, what is the tendency of having a diseased boy? 50%, because out of two boys, one boy is diseased. Now, X-linked recessive diseases mostly affect boys. In most of the cases, girls are carriers. Boys, mostly diseased. Girls, mostly carriers. Right, this is the same kind of inheritance pattern is seen in case of both hemophilia as well as color blindness. You have to remember this, and that's it. And nothing else will be asked in your examination. That is what you are supposed to know. Okay, just go through the crosses given in your book. You will be able to appreciate this jolly well, because we have done a lot about monohybrid cross, homozygous, heterozygous, etc., etc. Right. So with this, we conclude the chapter of genetics. There's nothing much to be taught in genetics. So I hope let's end the class over here today. So with this note, let's end the class over here today. Meet you with the new chapter soon. All right.